Now, some of the reactions that we've learned with amino acids or with carboxylic acids is the Helmholtz-Solinsky reaction. So in this reaction, we have an alpha bromination of a carboxylic acid. So imagine if we were to make an amino acid. So here's an amino acid. And one way that you could do that is use the Helmholtz-Solinsky reaction, make your alpha brominated compound, and then replace it with a nitrogen. So you could do a SN2 reaction. Might be a little bit slow because this is secondary, it's a little bit bulky, but it can work. So for practice, one way to do this reaction is to figure out what do you need as your R group, and then how do you use this reaction. So let's try it for leucine. So first I'm going to look up what leucine looks like. And leucine is a 1, 2, 3, 4. It's a isobutyl side chain. Okay, so here's your alpha carbon right here. You have a carboxyl group, an amino group, and then you've got this isobutyl group. Now notice amino acids also have chirality, so we usually talk about L amino acids. So all of these amino acids start off as L. But when we do these reactions, because it's SN2, it's backside attack, but this is racemic because here, this could attack on the top or the bottom. So it's very difficult to get chirality here that would be needed, but it's one way to make a racemic mixture of amino acids anyway. And this amino acid leucine, if we were to make that with this scheme, we would have to start with this carboxylic acid. And so this carboxylic acid will get alpha brominated, and then from there, we'll replace the bromine with an NH2. But the problem with this, again, it's that the bromine will get added on either wedge or dash. So I put a squiggly line there. Squiggly line means top or bottom. Okay, so that means that I'm going to lose my chirality in my amino acid. And so there are other kinds of purification methods that you could use to separate a racemic mixture. But we will just stop it here and say we will get a racemic mixture. All right, so a pretty simple procedure. So I need to put in the reagents then. So one, I use PBr3 and Br2 and water. And then my third step was I used ammonia. Do you guys know why I use an excess ammonia in this step right here? The hint is think of the mechanism. So here's the mechanism, and when this comes in to attack, we're going to end up with NH3 positive, right? So that's going to be protonated because we have 3 hydrogens here, so we'll have 3 hydrogens here. So we need another one to come and take that off. So one's just acting as a base to neutralize that for proton transfer. All right, and so similar question, but just working it backwards. If we did a starting material here, and we did PBr3, Br2, and we did H2O workup and excess ammonia. What amino acid would we get? So first, recognize COOH means carboxylic acid, C double bond O, OH. And then my NH3 is here, but it's racemic, so I'll do a squiggly line. And so this is the amino acid with a phenyl group, which some of you may already know is phenylalanine. Phenylalanine, by the way, is in aspartame, that artificial sweetener we were talking about. And people who have a hard time breaking that down can get phenylketonuria, which is another disorder. And so it's on the warning labels of aspartame or equal packets. Okay, we also learned a couple of other syntheses from previous chapter that you could use to make an amino acid. And so you could do a Malonic ester synthesis to get an R group. And then this is what we started with in these reactions, right? Carboxylic acids. We could then take a carboxylic acid and make it into this with the reaction that we just had. Another thing you could do is instead of starting with this, you could start with a modified version where you have a nitrogen group already on there. And then you could add an R group on right here. So using the same kind of step that we have here, you could add the R group on, and you could do the heat and hydrolysis to get the amino acid this way as well. Okay, so let's try doing that. So if we wanted to make isoleucine using this synthesis, so I'm going to start with amidomalonate to add an R group. So my sodium ethoxide, and then for each amino acid I have a different R group. So let's say valine. I haven't used valine in a while. When you look for valine, it's an isopropyl group. Okay, so isopropyl is secondary, so it's not the best, but it's okay to use it. If I had tertiary, I'd probably be in trouble though. Okay, because of the steric bulk. And once I have this, then I'll have my isopropyl group on there. And then from here, it's just a matter of hydrolysis. So one of these will turn into it'll turn into carboxylic acid. So I'll have H3O plus in heat. That'll turn this into a carboxylic acid and ultimately CO2. And then I'll have this molecule, and the acid also hydrolyzes this off, meaning I have H2O come in and let this go. So I'm going to end up with a free amine here, and this is going to be hydrolyzed to carboxylic acid also. So H2O will also attack here and let go of the OET group here. So it's a bit messy, but what I'm going to end up with is my carboxylic acid there, my nitrogen, NH2, and I'm going to end up with my isopropyl group. So there's my valine. All right, a peptide is when you link amino acids together. So we talked about some basic amino acids like glycine, alanine. And you can link them together because they all have a carboxy terminus and an amino terminus. And together, these can form a peptide bond, or we've been calling it an amide, right? An amido bond. So these are, peptide bonds are amides. Okay, so that is, so you think of the mechanism as sort of something like this, although that's not really what it's doing, but it might help you figure out how to link those together. So this is a new bond that becomes a peptide bond. And same thing with these guys. So this can sort of think of that as attaching here, and we lose water and we get this. So this is a kind of condensation reaction because we're losing water. The OH group and one of these H's forms water. All right, often we go from, see this free amino group here. So the free amino group here to the carboxy terminus here. That means I have the N terminus and the C terminus. So it's N, ala, gly, and then the C terminus. So that's the convention that's used in doing the directions. So I have N terminus on the left and then C terminus on the right. All right, so let's practice then. I want you to link some amino acids together. Let's link together the ones that form this 4-letter code. So C is cysteine, H is histidine, E is glutamic acid, and M is methionine. So find those amino acids and how would you link them together such that you have an N terminus and a C terminus like that. Go ahead and give that a try. And once you draw one of these separate ones, then think of lining up the next one so that the N terminus is here and the C terminus is here and the R group is hanging off of the molecule either down here or hanging off here like a charm bracelet sort of. Okay, so I would say let's line them up so that your amino and carboxy groups are sort of in the middle and your R groups are hanging off of it. Okay, all right, go ahead and give that a try. All right, so I think what you see is you've got the amino acid in red and it had an OH group, but that OH did a condensation with this nitrogen, so we lost water here. So I'm just going to highlight my peptide bonds. This is a new bond that formed between the two amino acids, the blue one and the red one. And then had another amide bond form here, so there's another linkage here between these two amino acids, and then I had another linkage here between those. So these amide bonds are the peptide bonds. And as you can imagine, they're pretty strong. I mean, we want to have strong bonds in our proteins, right? So if you think about it, amide is perfect because of resonance stabilization. That really helps to strengthen the peptide bonds here. But they're not so strong that you can't break them apart. So when we digest our food, that's what happens is the proteins break apart into amino acids. All right, so I want you to sort of think about this because we have these, the peptide chain or backbone is really made out of these amide bonds. And then notice that your side chains are sort of dangling off of that chain. And so that's got a lot of important consequences for protein structure. So let's talk about protein structure. Protein structure is how the amino acids are linked together and how the three-dimensional shape is built off of those structures. There's different levels of protein structure. One is just simply what amino acids are linked to what. So CHEM, for example, our little peptide here, that's an example of primary structure. I just want to know which amino acids are linked together. So that's a sequence. Secondary structure is how those strands might interact to make a 3D structure. So, for example, we talked about that peptide backbone, and then all the R groups hanging off of that backbone. Well, it turns out, if you look at it from the side, it turns out that due to the way that these side chains can interact, it's possible that because there might be some hydrogen bonding between different regions of these strands. So maybe we take a strand, right? And maybe this part of the strand might be attracted to this part of the strand because they're both polar and maybe they have an ability to hydrogen bond. So now as you look at the different regions, they might assemble themselves into a kind of coil or helix, and that is stabilized by these hydrogen bonds between different R groups. And that's why it's important to know which amino acid side chains are polar and which ones are nonpolar, and which ones can be charged or acidic or basic. That all has a consequence in terms of its 3D structure. So based on its pH and based on its charge, based on whether it can hydrogen bond, based on if it's hydrophobic, all of those kinds of qualities can lead to whether you have an alpha helix or not. So this is called an alpha helix. From the side view, it's sort of like a little coiled up wire, but that's a strand of amino acids like this, so that strand can coil on itself. If you look down from the top, so let's say you're looking down this way, then that's what it looks like, is it sort of looks coiled up like that with the R groups hanging out. And it's the hydrogen bonding that really helps to stabilize that alpha helix into that kind of structure. So that's one kind of secondary structure, is the alpha helix. And another very common one is called a beta sheet. If you look at amino acids, they have sort of this pleated quality to them. So that pleated quality comes from the one strand of one peptide and another peptide, and they're sort of hydrogen bonding like this across from each other, like that. Okay, so these kinds of hydrogen bonding can result in a pleated sheet. Like if you think of a pleated skirt, it's just basically zigzag. Okay, so that's another kind of protein structure as well. So with tertiary structure, often looks at secondary structure, but in a larger context. So let's say we have a much larger protein chain. Okay, so some regions might show secondary structure, like this is our alpha helix, so there's secondary structure within this. We also see some beta pleated sheets right here, so that's sort of hydrogen bonding between strands. Okay, we also can have lots of other kinds of interactions as well, not just localized, helices and pleated sheets, but we might have some regions of the molecule where we have hydrogen bonding. So let's look for all the hydrogen bonding interactions. So here are the regions for hydrogen bonding. So you can see some, the N-terminus of a peptide, and let's see, an alcohol group here, or you have an OH group of, say, a tyrosine residue, and you've got this carbonyl group somewhere, and you have another carbonyl and an amine. So you've got lots of potential for hydrogen bonding that ties different regions of the strand together. Okay, so you've got loops and turns and things that occur as a result of these hydrogen bonds. And then we also have things like this, which is hydrophobic regions of the molecule, like dissolves like. So nonpolar regions like other nonpolar regions. So they might also form types of attractions to each other. We just call those hydrophobic interactions. And then there are two other kinds of interactions that we often see. One is this kind of positive charge and negative charge. So that's often a consequence of the pH. We're in the pK of the side chains. So this is due to, this is what we call a salt bridge. And a salt bridge is where you have an ionic bond, or an ionic negative positive electrostatic attraction. And then the last kind is actually, I grew up in the 80s, and I remember getting my hair permed, or, always using a curling iron. But when you go to a hair salon and they perm your hair, the idea is that even when you wash it, it still curls up again. And so it's called a permanent. And what's happening there is, hair is made out of protein, and when they set chemicals into your hair, so they roll your hair up and they'll put chemicals onto it. What's happening is they're forming new disulfide linkages. And so these are called disulfide linkages. And remember back to which amino acids had sulfur. It was cysteine and methionine. And cysteine in particular is the one that has SH groups that get oxidized to form an SS linkage. So that's a kind of interaction as well that leads to tertiary structure, which is how parts of the chain interact with each other. Okay, the shape of the proteins is very important biologically. For the function of these proteins often relates. So for example, enzymes, it's really important that enzymes, they work in our body for a variety of functions, and the function relies on the shape and the structure. So if I ruin that structure, then I pretty much ruin the function as well. So for example, take hand sanitizer. When you use hand sanitizer, you have alcohol, and you also are disrupting proteins, presumably bacterial proteins, right? So you're destroying bacteria by destroying their proteins. We call that denaturation. You could do that by heat, so you could heat to destroy the protein shape. You could also use solvents like alcohol in a hand sanitizer, or you can use mechanical means, which is when you wash your hands, you can literally rub your hands together and really try to break up those proteins. Sometimes in the lab we use a sonicator, which uses ultrasound waves, and so you can try to break up proteins that way. Or pH changes also very drastically change the shape of a protein. As you can imagine, those linkages here, all of these charged interactions rely on the pH of the solution. So once you disrupt that, you can really change the shape of a protein. So here's an example of a nicely structured protein. We have an alpha helix and we have a beta pleated sheet. But once you apply heat or mechanical means or pH or solvent, you can really ruin the structure of it. It just unfolds or unravels and it changes. You've seen this happen probably when you cook eggs. You see the white parts turn white, and that's because the proteins are denaturing under heat. You also see it probably when you have milk that goes bad. So when milk goes bad because the bacteria take over, particularly lactic acid bacteria build up lactic acid, pH of your milk changes, and then the proteins coagulate or they precipitate out. So you can see chunks of milk protein at the bottom, and it smells really sour and bad when milk goes bad. You can also make cheese that way by precipitating the proteins early before all that acid takes over, right? You can heat up milk, and this is actually a great way of making paneer or fresh cheese. Is take a gallon of milk, add some vinegar to it, change the pH, heat it, and then the cheese will precipitate out, and you can squeeze out the water and with a cheesecloth, make some solid cheese out of it. It's really tasty. So, so that's actually a good example of how proteins denature. So I just remembered this experiment in high school I did, and actually in high school chemistry, I had no idea what was going on half the time, and it was only till I became a teacher that I understood. But one of the experiments we did was to heat milk with vinegar, and like I said, you can denature the proteins that way. And there are two main proteins in milk, the caseins and the whey proteins. And many of you are probably maybe familiar with protein powder, and sometimes they use whey from milk. And sometimes people are allergic to these proteins, not lactose intolerant. But anyway, these whey proteins stay dissolved because they're mostly soluble in water, and the curdles, or the curds, curdle, and they fall to the bottom of the beaker. So you could filter that out and get those solid curds. And maybe you know the nursery rhyme, Little Miss Muffet sat on her tuffet eating her curds and whey. So the curds, you can, which can be cheese, you can actually mix that with water and make a kind of glue out of it. So then we took our paper for our lab with our procedure and everything written out, and our teacher, Mr. Monaco, had us fold the paper in half and glue each of those halves together using that paste that we made from the curds. So then I kept that paper, and I still have it to this day. Finally, the last structure is quaternary structure. When you have some proteins that are pretty large, like hemoglobin, and I have a model of this in front of me, hemoglobin is actually composed of different kinds of units of tertiary structure. So maybe I have a strand of many amino acids here, and it turns out that this subunit and this subunit and this one get together, and they might form a greater structure. So for these larger proteins, they may have multiple subunits, 2 or more chains. This is an example of one with 4, so hence the name quaternary. But quaternary actually is referring to the fact that you have primary, secondary, tertiary, and then quaternary. So it doesn't have to have 4 chains. But hemoglobin is a classic example because each of these chains contains heme, and heme is a molecule that contains iron, and that's your transport of oxygen in your blood. So you've got different subunits here, and the way that they aggregate to form one more complex protein, that's called quaternary structure.